Welcome back, friend. Now we discuss about the next step of recombinant DNA technology, that is introduction of recombinant DNA into the suitable host cell and multiplication of gene. This entire process is called gene cloning. So in this process or in this step, what we do, so here the recombinant DNA that we made in a previous step, that we introduce into the our desired host cell. It may be the host cell may be prokaryotic or maybe eukaryotic, and multiplication of that DNA, nothing but a recombinant DNA in the cell. Okay, so both we are discussed here, and this process, entire process, gene cloning, and this is a multiplication of gene, nothing but a gene cloning. Okay, next is the gene transfer. So for the introduction of DNA into the recombinant DNA into the cell, nothing but a transfer of recombinant DNA. So the gene transfer, so there are mainly two processes of gene transfer. One is a natural method and another is a artificial method. So in a here we artificial, by using the artificial method, we introduce the recombinant DNA into the cell. Okay, so we in previous video we discussed about the list of recombinant list of this method, natural method and artificial method. In this video we discuss about some methods of artificial method of gene transfer that is used for the transfer of desired DNA into the host cell. So let's start. So number one process of gene transfer is the micro injection method, and this micro injection method is an effective method of gene transfer. So what we here do, so here we use a micro capillaries and microscopic device in a such a way a cell survive and proliferate in the culture. So here we use a micro capillaries and microscopic devices without killing the cell and without disturbing the culture. We use this without disturbing the culture even. So here during this process cell is not dying. Okay, so cell is survival. Next is, so here we use a two pipette, micro pipette and the holding pipette. So holding pipette is used to hold a cell, and micro pipette is used, it's just like an injection that introduce the desired DNA, deliver the desired DNA. Okay, next is, so in a 1 minute this 10 to 15 cell in 10 to 15 cell the desired gene or the recombinant DNA is introduced, and we can visualize on a screen by focusing the video camera to the microscope. You can see on a screen how this method is. Let's start the another method. So the another method of gene transfer is a biolistics. Only some method of gene transfer we discuss, and all of these is artificial. Okay, so biolistics method. So in a biolistics method, or is also called a micro projectile gun method or shotgun method. So here we use a gun, and you can see the picture of that gun. So in that gun we add the DNA coated with the micro metal like gold coated or tungsten coated DNA, and that DNA we place before a plastic. You can see that before that plastic. Okay, next is here two projectile system are used, micro projectile and macro projectile, so depending on the size of DNA. So in gun actually this gun contain what, so highly pressurized nitrogen is applied to the drive the micro projectile, nothing but our DNA. So with the help of high pressure of nitrogen, this our desired DNA is introduced into the tissue, integrated into the host DNA. You can see on a picture, you can see on a screen. Okay, next one is a use of polyethylene glycol. So what is this method is also this is artificial method. Yes, this is also the artificial method. Next one is about a polyethylene glycol method. Polyethylene glycol, so use of that to introduce desired DNA into the host cell. So applicable to the protoplast only. So what is protoplast? Cell without a cell wall is called a protoplast. Okay, so polyethylene glycol actually what do, and this is only used when the lysosome is used as a vector. Okay, so lysosome containing used to uptake the lysosome containing DNA, used to uptake the lysosome containing DNA. Next, this PEG actually what do? This PEG alter the surface charge of cytoplasmic membrane and protoplast, so stimulate endocytosis. So this liposome get bind to the cell plasma membrane and then introduce the DNA into the cell. Okay, so along with the 50% of PEG, Ca, calcium chloride, sucrose and glucose, all this also present in that solution. Not only PEG is successfully applied to the rice and maize plant. Okay, you can see on a picture, you can see on a screen. Okay, next one is the electroporation. So this electroporation method is also used to introduce a desired DNA into the host cell. So here we use a very small electric voltage, that is 4000 to 8000 volt per centimeter for a millisecond only. Okay, so we introduce the current into the solution of calcium chloride. So the bacteria and our recombinant DNA both are in a solution, and we apply the current or voltage about 4000 to 8000 volt per centimeter for millisecond, and due to this current, the what happened due to this current allow the membrane pore to open. So membrane pore get permeability of the cell, or we can say the membrane pore is size is increases due to this shock, and so our desired DNA get introduced into the host cell. Okay, to facilitate the cell to uptake the DNA by reversely altering the permeability of the cell. Okay, so pore size is gonna increase due to this and is used to efficient transformation. So here due to this the efficient transformation is takes place. You can see the on a screen how we can do that. Next is the fifth step of recombinant DNA technology, that is selection and identification of recombinant cell or transform cell. So after a desired gene is incorporated into the marker gene, now we know that what is meant by marker gene. Okay, on a marker gene is present on a plasmid or is present on a vector, and only in that marker gene we introduce the desired DNA by using the restriction enzyme. Then TETR, then this tetracycline resistance gene is inactivated. After the introducing the our desired DNA into the tetracycline resistant gene that present on a plasmid by using the restriction enzyme, then that marker gene is inactivated. Okay, so now so that inactivated and cell grow in a media containing ampicillin, not the tetracycline containing. So due to this marker gene or tetracyclic resistant gene is inactive, so if this our desired DNA, if this our recombinant DNA is present in the cell, then that cell can grow only in a media containing ampicillin, not in a media containing tetracycline. Okay, so if the cell is grown in a tetracycline ampicillin containing media only, then that cell is a transformed cell or recombinant cell. Okay, so that the way we select or we identify the cell. We can see now in detail. In recombinant, the rDNA is insert. So in a recombinant cell, rDNA inherit as its own parts of genetic material. So that recombinant DNA become its own part. So this foreign DNA is become the own part Okay, so this foreign DNA is become the own part of this bacterial DNA, so that also multiply with the bacterial DNA and also express, not only incorporate, also express. So after introducing of desired DNA or recombinant DNA into the cell, we get a three cell in the media. Number one, the non-recombinant, some are recombinant and some are unaltered plasmid. So in a some are recombinant means in them our desired DNA is present and that also express. Non-recombinant, so our desired gene is not introduced into the cell, or that cell is not produced, even present, but that cell is not produce that cell. Okay, so produce our desired product. Next, unaltered plasmid. So plasmid as it is present in the cell, that not do anything, just as it is, as a waste one, as a waste we can say. So you can see here, this is a this is a plasmid example of vector, and here our tetracycline resistant gene is this is, and in this gene we introduce the our foreign DNA into this, at this site, at marker gene, and we get a recombinant DNA. This is a recombinant DNA, and this recombinant DNA, with the help of our previous method of gene transfer, we can introduce this desired DNA, this recombinant DNA into the bacterial cell. So this recombinant DNA become the parts of this own bacterial DNA, and that cell is called recombinant cell and also express. So when this cell is grow on a media containing ampicillin only, then they can grow. So they grow. This is a colony of these bacteria that grow, and that all are recombinant or not, that we want to see now. So for that we replica of this. So with the help of this replica of these colonies, we are plated on the another plate containing tetracycline colony, and so we get these two pure recombinant. This is to the pure recombinant cells colony. Okay, so this is all about so this is our recombinant cell. Okay, now we next see, now we get a recombinant cell. Now we want our product. So how to get a product? So our product, suppose the insulin or protein, any protein. So how to get that? So bacteria contain the many proteins and micromolecules. Separation from the other protein and micromolecule. So our protein is present in the bacterial cell. So this is a bacterial cell, and our protein is present in the bacterial cell. This is our protein. This is shown by a red. Okay, so this is our protein in the bacterial cell, and this bacterial cell we burst this bacterial cell or lyse, and after lysing we introduce this into this column, and after introducing this column, or this is nothing but affinity chromatography, used to separate the component of the cell, used to separate our desired product. So in this the antibodies is present in the column, and that bind to the target protein, and protein is released in that. After that we get a pure protein. So by using the affinity chromatography we obtain our desired product. Okay, this is one of the method. Depending on a product, we different method we are uses. Okay, so this is the steps of recombinant DNA technology. I hope this is helpful for you. In next we are discuss about the somatic cell hybridization. Okay,